Hi guys, welcome to Math Skills on section 7. So this is my last video on the math skills, because if you are looking now through the specification, there are actually no any requirements in terms of the maths for section 8. Hence, I'm not recording any videos for that section. So let's get started. One of the information that you need to be aware of is the ratios in terms of the monohybrid and dihybrid crosses. But I would strongly suggest you also memorize the ratios for other crosses, because that would be really useful for you to work out the predicted and observed values and recognize the crosses that you are dealing with at the specific moment. So monohybrid for the second generation, we've got the ratio of 3:1, dihybrid 9:3:3:1, codominant 1:2:1, and epistasis 9:3:4. So I would strongly recommend you remember those ratios. They will be useful in terms of the past paper questions and the problem solving questions. Another skill that we've got here, you need to show understanding of the probability associated with inheritance. So here, please watch the video on the statistics. Everything about the probability and 5% value has been described there. But we're going to have a look here at the different types of the questions when we can then use our ratios and come up with some predictions. So here we had the past paper question that shows you the phenotypes and the number of offspring produced using those information. So we've got the phenotype and the offspring, and we've got the number of the offspring. So as we can see, the tall plants and normal leaves. So tall plants, okay, are dominant T, okay, allele shows the tall plants, and dominant allele M shows the normal leaves, which are linked here, are high in the number, but also the draft plants and mottle leaves are high in the number as well. So what is our explanation for this? The high number identifies that those alleles has been linked. The lower number in here could be the explanation of those could be the process of crossing over. So in this situation, getting the numbers that are far away from the ratio, what we've mentioned for the dihybrid cross, which is 9:3:3:1, will suggest that those genes, those alleles are linked. Okay, so we are dealing here with the autosomal linkage. But the question is asking you to complete table 2 and show the expected ratio of phenotypes if the same cross has been carried out, but the genes were on different homologous pairs of chromosomes. Other words, they are not linked. So to remember the ratio, that would be then a normal dihybrid cross. So the ratio for this will be 9:3:3:1. And then the phenotypes will stay the same but different ratio. Hence, I would strongly recommend you do remember those 4 ratios that we've mentioned about at the beginning of the video. So another question here, we've got the epistasis, really lovely. It shows you the interactions of 2 genes, and what do you really need to remember is that from the white, okay, we can get to the green only if the recessive homozygous it's presented. Here, if we had the capitals, okay, so dominant alleles or heterozygous, it won't happen. But if from the green to yellow, we can only get if we've got at least one of the dominant alleles, B, which stands for the which codes for enzymes B. But also what we've got here, big A shows you the inhibition. So if that's there, inhibition is taking place, and the recessive homozygous will produce non-functional enzyme. Hence, we're not going to get a yellow color. So we need to complete the genetic diagram to show the possible genotypes and the ratio of phenotypes expected in the offspring of this cross. So what are the I can't write on this because I can't hide this tool. I'm not sure how, but let me just open the Word document if I can. Right, and we can do it in the Word. Right, there we go, smaller. Right, so that will be our working box here. Okay, so the gametes that will be produced by first parent, it's only the recessive A and the recessive B. We don't have any more combinations, but when you're looking at this heterozygous here, we will have plenty combinations of the alleles. So we could, starting with the capital dominant A, those are two possibilities, and with the recessive A, those are another two possibilities. So this is the gametes that will be produced by the second parent. So then what you need to do, you need to obviously do a Punnett square. So let me insert the table. So we will need, right, there we go. So here I will put the gametes of the first parent, and there I will put the gametes of the second parent. Right, A and B. So let's draw. Correct. Let's join them together. So this is the fertilization now that we are talking about. Those are possibilities of the offspring. Right, and here A, B, B. Right, sorted. So those are the genotypes. Okay, genotypes. But what is the next question? They're asking you about the phenotypes. So phenotypes, it's then something that we can read off from here. So what will be then this fruit color? So we've got the inhibition. So we've got big A. So it's obviously not going to be green at all because we've got big A. So it stays white. Same will be that one big A, it's enough to inhibit this process. Never mind, this is not going to take place because green it's not present, so that's white. Then we've got lower cases a's, so we're definitely getting a green color, but we've got one dominant B, so that is going to express the enzyme 2. Hence in here we will have the yellow color. And finally in there we've got two recessive A's, which is good, it's gonna give us green, but we've got two recessive B's, which produces non-functional enzyme. We cannot get yellow, hence we're staying on green. Right, sorted. So those are the phenotypes of the offspring, and the ratio of the phenotypes would be then 2, which is white, okay, to 1, which is yellow, to 1, which is green. Okay, and that's the whole working out for this question produced. Okay, so remember there were the gametes, then you're doing a Punnett square, then you're working out the phenotypes, and they wanted the ratio of the phenotypes, right? So this is then the answer that we're getting, exactly the same as ours. Right, another skills here. This is the high score test and the significance level. So please go to section 7 video and watch the video on the high score test. We don't need to repeat again. Another skill is the Hardy-Weinberg equation. That's again in section 7 videos with plenty examples, all of the possible examples. So make sure you watch that. And the final one is to look at the knowledge of sampling to the concept of the genetic drift. So again, you can look at this in section 7 videos, right? And log scale. Again, we've done that. So the log scale we are going to use when there is a big range in the numbers. So a log scale, it's going to be amazing for this representation. So that's everything in terms of the map skills for section 7. The final ones, as I've mentioned, are already covered in the videos. So please go away and watch the videos if you are not sure about those skills. Right, so that's everything for the map skills. See you later.